Whenever we are learning a biology technique, we have to keep one thing in our mind, that is, after learning that particular technique, are we able to analyze the data? Are we able to understand the basic principle as well as the results of that particular technique, right? Because in today's time, we are not requiring the technicians who can run the instrument. We are actually requiring the people who can design the experiment. And only those people can design the protocols and experiment who are actually understanding both basic principle as well as data analysis as well as result analysis. By keeping this fact in our mind, today we are going to understand analysis of apoptosis by Annexin-V PI, Annexin-V propydium iodide flow cytometry. We'll understand the basic principle, we'll understand its graph, and we'll see one of the examples also. So before starting the analysis of apoptosis by flow cytometry, we will first understand what is flow cytometry, what is the basic principle of flow cytometry, and how a flow cytometer is working, right? So let's start with basic principle of flow cytometry. The word cytometry is used when we are measuring physical and chemical properties of single cell. So what are the physical properties, what are the chemical properties, what are the proteins present in that particular single cell? When we are doing, we are calling the whole thing as cytometry. And when I am adding the word flow in front of cytometry, then the flow cytometry is defined by the statement that we are measuring the physical and chemical properties of single cell, but we are using a fluid system or fluid stream in an instrument called as flow cytometer. So only one thing which I am adding here is a stream of fluid. So let's understand what I mean by the fluid system, or what a flow cytometer is actually doing when we are putting our cells in the instrument. So if I look at the functional aspect, of the instrument is very simple. It is taking single cell suspension, and by using the pressure of fluid, so it is the flow of fluid, and by putting or by applying the pressure of this fluid, the instrument is converting these bunch of cells in a system where one cell is passing at a time. So it's very simple. There are so many cells. You apply the liquid pressure from both the sides. So you can create an array, or you can create a stream of buffer or stream of a liquid where one cell at a time is passing through. And that one cell is interacting with laser, the light source. And these interactions are measured by different kind of detectors. The principle is very simple. And this application of pressure and generating this stream is called as hydrodynamic focusing. Very simple to understand. You take some cells, you apply hydrodynamic focusing, you apply the flow of liquid, a pressure, create a single array of cells. Then one cell at a time is interacting with laser and giving us some information which is detected by different type of detectors, right? Nowadays there are flow cytometers which are coming with acoustic sound waves, where along with hydrodynamic focusing, a sound wave is also being utilized to create this array of cell, or to create this situation. So the principle is not in the working of instrument, but principle is lying mainly where we have to understand what kind of interactions are happening between the laser and the cell. So let's understand that. Let's understand the interaction of light with the cell. So here it is our light source, the laser, Then we are having incident light beam, and here is our single cell. So if the cell is not coming, then the laser is easily passing through the liquid because there will be only buffer, there will be no cell, and it is detected by detector as no event. And if a cell is there, then the cell will interact with light, the light will be scattered by this cell, and the machine is counting it as one event. Second cell means second event, third cell means third event. It is going like that. So absence of cell, no events, and presence of cell, yes, there are events. Now, the light can interact with the cell in three manners. The first manner is the light can scatter from the edges of the cell, and the angle of this scattering is between 0.5 to 5 degrees, and we call it as what? Forward scatter. What do you mean by forward scatter? The light is coming, interacting with the cell, scattered between the angles of 0.5 to 5, and this scattering is occurring because of the edges of cells, right? We are calling it as what? Forward scatter. And the forward scatter is giving us information about the size of cell. So if we are having bigger cell, then of course the scattering will be higher. The value of forward scatter or FSC will be higher. So if the instrument is telling us that for a particular event FSC value is this, that means that is the indicator of cell size. Higher the cell size, bigger the cell size, the value of FSC is high. Now, some of the light can enter inside the cell and it will be scattered by the organelles, by the biomolecules, or you can say by the granules of the cells. This kind of scattering is between 15 degree to 150 degrees, and we call it as what? Side scatter or SSC. And we already know now that the value of SSC is giving us information about internal granularity of the cell. So more granules, higher the value of what? Side scatter. Bigger the size, higher the value of what? FSC or forward scatter. So the FSC and SSC, two parameters, forward scatter and side scatter, are giving us the information about size of the cell as well as granularity of the cell. The third information which we can get from a flow cytometer, if my cell is containing a fluorescent protein like green fluorescent protein, red fluorescent protein, or stained by a fluorescent dye like propydium iodide, cypergreen, cytox, or the proteins of this cell are labeled with fluorescently tagged antibodies. So it can have inherent fluorescence, it can have presence of a fluorescent protein, it can be stained by a fluorescent dye, or it can be having fluorescently labeled antibodies which are binding against specific proteins of this cell. So all these type of fluorescent molecules can absorb the light at a particular wavelength, and then emission of light will happen, and then ultimately we will get the detection or we will get the information. So three types of information. First, forward scatter size, side scattered granularity, and finally presence or absence of a fluorescent molecule, right? So guys, let's start now understanding how we can use a flow cytometer to analyze the apoptosis. And before going into technicalities, let's just understand what is apoptosis. So apoptosis is a kind of mechanism of cell death where a healthy cell first undergo with mild convolution, means cells are going to shrink themselves. The chromatin will start compaction, means chromatin will decrease in size, or you can say ultimately there will be condensation of the cytoplasm. In the next stage, there will be breaking of nuclear envelope, nuclear membrane will be gone, nucleus will convert into small fragments, and cellular fragmentation will also start. and these small cellular fragments will be ultimately called as apoptotic bodies. Once small apoptotic bodies will be forming, then the nearby phagocytes will come and will do the phagocytosis of these small apoptotic bodies, and our cell will be completely gone. So this is the basic flow of apoptosis, where so many proteins and so many different molecular mechanisms like intrinsic pathway, extrinsic pathways are working. But right now we are not interested in learning the cell signaling and pathways of apoptosis. We are more interested how we can use these symptoms of apoptosis to analyze the apoptosis by the technique called as flow cytometry. So one of the symptoms during this process of apoptosis is the flipping of phosphatidylserine. Now what is that? So if we are having a normal cell or healthy cell, then the phosphatidylserine molecule in these normal cells is actually present the inner side of the plasma membrane, or you can say inner surface of the plasma membrane. But once the process of apoptosis starts, then these phosphatidylserine molecules will be flipped, and now they will be on the outer surface or outer leaflet of the plasma membrane. And finally, at the end of the apoptosis, there will be membrane disintegration. So the membrane will become permeable for outer substance to come in. So we are working on two major symptoms of apoptosis. The first is initially the flipping of phosphatidylserine, and finally the increased permeability of that particular cell. So these two symptoms we will be utilizing to understand the apoptosis by using flow cytometry. Now how to do that? There is a protein which is called as Annexin-V. What is the specialty of that protein? The specialty of that protein is it can bind to phosphatidylserine. Whenever it is near the phosphatidylserine, it will show higher affinity and it will do a binding. And then second, we are going to use a dye, DNA binding dye, which is called as propydium iodide. What is the specialty of this dye? It can enter into the cells which are permeable. In healthy cells, it will not enter. In permeable cells, it will enter. So we'll take our cells in a test tube, in a tube. There we will add Annexin-V, which is fluorescently labeled, so we can get fluorescent signal, and we'll add the fluorescent dye, propydium iodide. What will happen? In healthy cells, nothing will happen because Annexin-V, which is present in the solution, cannot bind to phosphatidylserine because phosphatidylserine is present in the inner leaflet of the cell membrane. So phosphatidylserine is not exposed to Annexin-V, so there will be no Annexin-V binding, right? The membrane is integral. There is no permeability for propydium iodide. So the propydium iodide staining will not happen because propydium iodide will not be able to go inside the cell. Now once the apoptosis starts, the membrane is still having good integrity. So the PI staining will not occur because propydium iodide cannot go inside the live cell. It can go inside only in the permeable cell. But there will be positive fluorescence for Annexin-V binding. Reason? Because the phosphatidylserine, which was present in the inner leaflet of the cell membrane in healthy cells, now it is flipped and came out, and now it is exposed for the binding of Annexin-V. So the cells which are doing early apoptosis will be positive for the binding of Annexin-V and will be negative for the staining of propydium iodide. And finally, when the whole thing is finished, then the cells will be positive for Annexin-V because it is flipped and it is present on the outer side, as well as the cells will also be positively stained for propydium iodide because now the cells are what? Permeable. Propydium iodide can go inside, can bind to DNA and can give us the positive signal. So three types of cells can be there in a mixed population in our tube: healthy cells, no binding, no staining. Early apoptotic cell, then Annexin-5 binding, but no staining with propydium iodide. And finally, dead cells binding with Annexin-5 as well as positive staining for propydium iodide. Now, if we run the whole sample in our flow cytometer, then we get this type of graph. So you can see that on x-axis we are having Annexin-5 fluorescence intensity. On y-axis we are having propydium iodide fluorescence intensity. So these are the cells which are in this quadrant, lower left quadrant, are negative for both. Negative for both means they are not giving fluorescence for Annexin-5 as well as they are not giving fluorescence for propydium iodide. So which are these cells? Of course, healthy cells, because no binding with Annexin-5 as well as no staining with propydium iodide. Now these are the cells are positive for Annexin-5 but negative for propydium iodide. This quadrant, that is lower right quadrant. So positive for Annexin-5 means they have entered into the process of apoptosis, but still they are not dead because they are negative for propydium iodide. So we are going to call them as what? Early apoptotic cells. Now if I look at these cells, then these cells are positive for Annexin-5 as well as they are positive for what? Propydium iodide. So these are dead cells as well as positive. So they are late apoptotic cells where the apoptosis is done, or they are necrotic cells. So here there is a small confusion. Some people don't understand, or some people are having confusion that why these cells will also be containing necrotic cell. The answer is simple, because if the Annexin-5 is binding to these cells as well as propydium iodide is also binding with these cells, means propydium iodide positive cells are permeable. So the Annexin-5 can go inside the cell also and can bind to phosphatidylserine, as well as it can bind to cells which are having phosphatidylserine outer side or exposed to the outer environment. So we cannot differentiate whether these cells are dead because of apoptosis or they are dead because of necrosis, because Annexin-PI staining is telling us that PI is entering, so cells are permeable. So if the cells are permeable for PI, they will be also permeable for Annexin-5. So if there is permeability for Annexin-5, means Annexin-5 can bind to phosphatidylserine which are present inside the cell as well as which are present outside of the cell. So there is no method or there is no way by which we can define with this graph that these cells are late apoptotic or necrotic. That is why we are mentioning both. So healthy cells, double negative. Early apoptotic cell, positive for Annexin-5, negative for propydium iodide. And late apoptotic cell or necrotic cells are positive for both, positive for PI as well as positive for Annexin-5. So this kind of graph we. So now to show the example, the lab example, I am utilizing the data of our own lab. So there we have treated the cancer cells with an anti-cancer drug which is inducing apoptosis. So here we are having no treatment, or you can say at 0 hours. So most of the cells are negative for both. So most of the cells, 94.30% cells are what? Healthy. Some cells are early apoptotic, or some cells are showing necrosis or apoptosis. But after 24 hours, you can see that the cells are moving in this direction. It means there are cells in early apoptosis, that is 13.68. Healthy cell percentage is decreasing, and there are also the cells which are double positive, that is 7.27%. And if we look the same thing after 48 hours, then you can say that many of the cells are going under the process of apoptosis and many of the cells are already dead. So the healthy percentage, you can say from control we have reached from 94.30 to 37.96%, and there is cells, there are cells which are doing apoptosis as well as they are dead, and this death can be both necrotic as well as apoptotic, because I have already told you we don't have any way to find out here whether the cell death here in this compartment is occurring because of apoptosis or occurring because of necrosis. So this kind of data we get, where we can calculate the percentage of cells which are doing apoptosis, which are healthy, or which are doing necrosis or apoptosis. So I hope you guys have understood this video or this lecture, where we have utilized flow cytometry to understand the apoptosis. So guys, please subscribe and share our videos. You can always follow us on LinkedIn, Facebook. If you are having any doubts, you can WhatsApp us, you can mail us, and if you want to learn flow cytometry with us, you are always welcome. And stay tuned, because we are going to create more such videos where different applications of different biological techniques will be there. So thank you guys. Thank you very much.